Okay, let us talk. We're going to begin. This is a video we're going to talk about smooth muscle. So, smooth muscle. So in skeletal muscle, you have these long single muscle fibers that are then, grouped into, if I remember correctly, fascicles, and then that's blown up into larger portions of those. So that's what that is, and then that's what that is. So skeletal muscle, this is its normal anatomy. Smooth muscle does not work like that. It's not in nice little smooth ring compartments. So if we were to draw like a muscle belly for smooth muscle, so if in skeletal muscle you've got these short and then you've got these repeating discs of the sarcomeres. That's a quick hand sarcomere. Let me try to avoid the light getting in the way. Okay. So within skeletal smooth muscle, sarcomeres do exist. That's going to be my sarcomere. So they're all over the place. They're not in any kind of consistent arrangement within the smooth muscle. So they're kind of attached just by little filaments. So they're all over, and when they contract, they pull everything together so they constrict the smooth muscle. And so there are two types of smooth muscle. There's single, and so when you have calcium flow that causes the muscle, so the sarcomeres to contract. So if we look at single one, this is going to be my quick drawing of the sarcomere. So there's a quick drawing of a sarcomere. I've got the end of one and then beginning of another. What happens is for single units, between the two muscle fibers, we have what's called gap junctions. And so at gap junctions, they allow ions to flow completely through, no stop. They don't have to reach any kind of impulse or receptors on the other sides. They just flow freely from this side to this side. So if I've got lots of calcium in here, that's causing this specific muscle fiber to contract, it will cause this other muscle fiber to contract. And so that's really important for things like peristalsis, which is the movement of the smooth muscle in your bowels. So it's kind of like this consistent contracting that slowly moves food up. So one contracts, it'll just cause everything else to contract. So that's your single unit smooth muscle fiber. Make sense? Great. Your multi-unit ones, multi-unit. So say we've got here, there's one. So there's one, and then here's another. There's another sarcomere. They don't have those gap junctions. And so in order for both of these to contract, both have to have an influx of calcium. And that's important for other smooth muscle cells, like the muscles around the hairs on your skin. So that way you can contract some of them, but not all of them. So that's kind of like how you can get the hair to raise up all the way, and then it lets it slowly go down. Or it can, it can stay at certain points, or be all the way down, so forth. Makes sense? So, difference between multi-unit and single unit is single unit, everything just contracts all at once, whereas within multi-unit, each fiber contracts on its own, requires its own influx of calcium.
Reasonable.
Okay. We're going to talk about the two types of contraction. I'm going to draw fun diagrams. If you watch the first video, these diagrams will make a lot of sense. So there's this, there's that, here's this. Great. And there's my little phosphate. So this is that little zigzag. This is kind of like the base for the molecules. This is the myosin head. Here's our actin filament. This is a little phosphate group, which leaves the myosin head in what's called the cocked position. So it's like ready to attach and to pull the muscle and to contract it. So it starts out similar to skeletal muscle, and then what happens is we have what's called MLCK, or myosin light chain kinase.
Or milk.
Well, when I saw it, I first thought like Martin Luther King. And so the really interesting thing, another important difference between smooth muscle and skeletal muscle. Skeletal muscle has, so both have a fun little double helix thing. And so that little double helix is called tropomyosin. So that binds to the actin filament that is. And in skeletal muscle, you also have another thing that sits on top of that called troponin. And so troponin, when you have calcium concentration really high within the cell, it moves the tropomyosin out of the way, allows the myosin head to bind. The problem is in smooth muscle, you don't have troponin. So what happens is MLCK will add a phosphate. So we'll add a second phosphate onto the myosin head. So you have one phosphate and then a second phosphate. And what that does is it essentially makes it so the myosin head becomes more like preferentially bindy to the actin filament than the tropomyosin. So it just kind of like shoves the tropomyosin out of the way and allows it to bind. And then what happens is you have leaving, so you've got heat that leaves because your ADP, which is right here, so that's your ADP. Actually, I will write that in a different color. We'll do this one in red, and I will actually do this phosphate in a different color. And this one. So that's phosphates in red. Here's our ADP. I'm gonna make this. There's our phosphate, and then there's our ADP. And this phosphate, I'm gonna make blue. Okay. So what happens is then this leaves. Cool. So then the next set. So we now have those leave. So we're now left with our myosin head still bound. And we have that. And then what happens is super exciting. So these leave. This is, oh, I need to draw that a little differently. I have to show that it is now in the uncocked position. Those move. When those leave, that causes a conformational change in the myosin head, pulls it in. Oh boy, okay, let me, that doesn't really help much.
You have a lot of space elsewhere on the board.
I know. Gosh dang it, it's fine. I'm gonna have to try to pay really close attention to where those are. Okay, so there's that. I'm going to redraw this so it's not in a glare. I draw just right below it. Right, that's good. And then we can see this. I will draw my little phosphate. You can see that. Okay, if we shift this up just a tad, or down a tad. Okay, good enough for me. You can kind of see it. There's that. So then I'll go to this. MLCK will add a phosphate. In actuality, what MLCK does is it takes an ATP, breaks off a phosphate from it, sticks it onto this, and then the ADP kind of just floats off into the cell and does its ADP things. The body has a whole bunch of stuff that it does with ADP, and I won't get into that. Okay, so then what happens here? Okay, just to make sure that we're good. So we've got this, here's that, here's this. What happens is ATP will then bind to the myosin head. So there's a
ATP now?
ATP, so
ATP, okay.
yeah, so you've got 3 phosphates on it. And that causes the myosin head to unlatch from that and to go back into its uncocked position. So I'm just going to draw that. And then there are 2 things that can happen. So depending on the concentration of calcium within the cell, is it can go this way, hydrolyze ATP. So on the myosin head, it has part of the protein gives it the ability to hydrolyze the ATP and break it into ADP and a phosphate group, basically getting it prepped again to cock. And so this can help like reduce the amount of energy that is required for the smooth muscle. So this is really nice for things like bowel movement, where you want consistent, but doesn't have to be super fast, doesn't need to be super high energy intensive. Or what can happen, I need to draw my ADP on there. That's an ADP. What can happen is we can use this, and we'll use an enzyme called MLCP. And what MLCP does is it removes this phosphate. So we get phosphate goes away, and ATP turns into ADP plus phosphate. So either step we go, it still hydrolyzes the ATP, but in this step, it removes this second one. So the nice thing about just skipping here is it allows us to go through the contraction quicker, quote unquote. For skeletal muscle,
like
it goes,
skipped or something?
yeah, so it skips this step.
Okay, yeah.
So for skeletal muscle, it will go through like the steps really, really, really quickly. But for this is called tonic contraction. Smooth muscle that goes through tonic contraction goes through the cycle, but slower. So it ends up using overall less energy.
Alright.
Okay. Now we're going to talk about phasic contraction. Okay, I want to make sure that I get these in places where they're not in the way. Okay, what are we doing on time? 13 minutes, 12:19. Okay. So we got this. Here's that. There's your phosphate. Woohoo! Okay. So currently sitting in its cocked position, we have MLCK. Right, and then I'll go here. Happens MLCK adds that phosphate group, allows the myosin head to bind. Right? No, not quite yet. Sticks it on. This goes on. This is there. Ready to go, and then happens. We have ADP that leaves, phosphate leaves, and we have heat. I go here, attaches. But the weird thing is we also have MLCP in here, also pulling off that extra phosphate. So now this head is kind of just stuck in the uncocked position and contracted. So then what happens, make sure I'm not getting into any glares. Perfect. So we have this, here's that. And then you have the myosin head just kind of chilling. And you have an ATP that's trying to bind to the myosin head. But because the myosin head doesn't have any phosphates on it, and it's in that cocked, well, it's in an uncocked position, it's really, really hard for the ATP to actually bind to it. So this is what we call the, oh, not lagging, latch. Latch phase. So this is how like sphincters work. So, because you want them to stay contracted for really long periods of time. And so that long period of time happens between this step and that step, where the ATP is just like trying to bind but can't. And so it just stays contracted for a long time until eventually the ATP binds and then is hydrolyzed into ADP and a phosphate. And so this is your phasic contraction. And the big difference between the tonic and phasic is when MLCP, which is called, that stands for myosin light chain phosphatase, which just, cuts off that phosphate, changes, the difference between tonic and phasic is when MLCP is used. Okay. Going to draw a muscle cell. Cool, here's our muscle cell. Actually, we're just gonna draw a line that way. Here's our muscle cell. Cool, you can see it decently well enough. So we have, actually we'll put it up here. Up here, there's some neuron, releases stuff. Acetylcholine tries to get this to activate. That causes voltage-gated sodium to come in, ends up causing calcium. Actually, no, it does not cause calcium to come in. This will, not sodium. This activates a G protein that activates what's called IP3. And so what IP3 does is it increases calmodulin. So calmodulin is, gosh darn that stuff. Haha, that's right, I can move this. Okay, so just know, you see it, I see it. So calmodulin is kind of like this little interesting protein, and it's got 4 binding sites for calcium. And when it's bound to calcium, so IP3 causes calmodulin to be activated because it causes, so SR, sarcoplasmic reticulum, opens up channels in this that allow calcium to flow out. So calcium flowing out goes onto this calmodulin. Our MLCK is naturally in an inactive state. So the way we like to describe it, it's kind of like this. And it's got, they call it foot and mouth conformation. So currently this can't do anything because its active site is blocked. So when calmodulin binds to it, we're going to draw our little calmodulin beam there. When calmodulin binds this becomes active, and this can then start putting those phosphates onto the myosin heads, which allow them to contract, and you can go through that tonic or phasic contraction in smooth muscle. And that's all dependent on this release, IP3, increasing, which causes a release of calcium from the sarcoplasmic reticulum, which is the endoplasmic reticulum in muscle cells. Calcium increases inside, calmodulin is bound, this happens. Calcium also causes, so the other one that we have, I'm just going to draw like that. So this is our, I will label this MLCK. This is MLCP. MLCP. And so normally within the cell, it is phosphorylated. So it has a phosphate group, and this keeps it in the inactive state, and that's because of a protein called rho kinase. When you have an increase of calcium, calcium will bind to rho kinase and deactivate it, which means because rho kinase isn't active, it does not put these phosphate groups on MLCP, which allows MLCP to be in its active state. We're going to draw it like that. Looks kind of funky.
It's Pac-Man.
It's Pac-Man, yeah. So when calcium concentration inside the cell increases, both MLCK and MLCP activity start going up. And so this adds the phosphate onto the myosin head, this removes the phosphate. Okay. Good so far?
Yes.
Great. Okay. Next thing, regulation versus skeletal muscle. So those are just largely determined based off of calcium concentrations. One of the interesting things, so in skeletal muscle, you have these T tubules. So here's our SR. This is a T tubule. This right here is what's called, this is a DHP channel. This is our ryanodine channel. When you have voltage change across the membrane of the muscle cell, this channel will open and will cause this channel to open. So we have calcium coming in from there and there. But in skeletal muscle, it's mostly just there. So this is skeletal muscle. In smooth muscle, you don't have a T tubule. So it's all based off of calcium concentration coming within the cell, either from voltage-gated calcium channels, or being released from the sarcoplasmic reticulum.
I see.
And so there are DHP channels on the smooth muscle membrane, also called I don't actually remember what that one's called. But it's not connected like the two channels were in smooth muscle. And so that allows smooth muscle to use the high concentration in the ECF and the high concentration of calcium inside the sarcoplasmic reticulum. So you flood the inside of the muscle cell. And that's how the regulation works versus skeletal muscle. Tonic and phasic are interesting. When you have tonic contraction, when you have rush of calcium, you have high activity of the MLCK, which puts the phosphate on the myosin head, and then you have a slow buildup of the activity of the MLCP, which removes that phosphate. So you usually go through 2, 3, 4, and then you skip back to 2, more often than you go all the way up to 1. Whereas in phasic contraction, you have high increase of MLCK and a really high increase of MLCP, and you can control that by how much calcium you pump into the cell. So if you put in a ton of calcium, you'll activate MLCK and MLCP. So you'll end up hitting the MLCP in that second step for the phasic contraction. So you'll add that phosphate group, and then you'll remove that phosphate group, and you'll cause it to more likely stay in that latch stage for a longer time.
Okay.
And that's smooth muscle.